So this typical diagram represents a very specific chemical message. What it is a saturation curve. Now you can do this in the lab for any chemical. You basically take the chemical, in this case hemoglobin, and you set up controlled conditions that range from 0 as the partial pressure of oxygen up to, in this case, 100. That's not a percent. You could keep going. But this is a really good estimate of sort of a maximum of partial pressure for oxygen in our actual living conditions. The idea here is a central curve, which we call normal. Excuse me, I don't have the pointer going. This curve, the blue curve, is an at-rest, normal CO2, hydrogen ion, and normal temperature. So you can see that as we increase the partial pressure of oxygen, we increase the saturation of the hemoglobin molecule. Now, because hemoglobin is a tetramer, 4 chains, each chain can bind 1 oxygen molecule. 25% saturation at this point right here is meaning that on average, the hemoglobin molecules in your sample are binding 1 out of the 4 sites. At this point right here, 25%. 50%, that's binding 2 out of 4. 75%, you're binding 3. And you'll notice we really flatten out up here. Now what's happening is after you get to 3, and you get there pretty quickly, partial pressure of 40 millimeters of mercury, that's a relatively low concentration of oxygen comparing to the gas exchange that we're doing. So we can easily get that into the lungs. So what happens after that? We start adding that 4th oxygen per hemoglobin. And the more we add it, when we get up here to 60, so we've already increased it 50%, and yet we're only at 90% saturation. You'll notice that as we keep increasing it, this curve is flattening out so that achieving a 100% saturation, or all molecules of hemoglobin carrying 4 oxygens, is very, very elusive. It doesn't actually occur. But from this normal curve, where we establish the baseline for comparison, what happens when we start exercising? When we start exercising, the CO2 increases. Increase in CO2 means increase in carbonic acid, increase in bicarbonate, and that means an increase in hydrogen ion and a drop of pH. When we exercise, it also means an increase in temperature. So the purple line actually represents the conditions of exertion compared to rest. Not in this experiment, but in asking that question, when you go into exertion compared to rest, what happens? What happens to the saturation kinetics of hemoglobin? What you'll see is that it goes from being 74% saturated down to about 66% saturated. That means that hemoglobin under those conditions is going to release more and more and more oxygen. That means greater oxygen delivery. And that's sort of where we left off last time. From this basic curve, looking at those conditions of oxygen transport by hemoglobin as they're established in the lab. So we see here the temperature curve only, so that at our 38-degree body temperature, you'll notice that around 40, we go from about 80 to about 60. So we're releasing 20% more oxygen when we increase the body temperature by 5 degrees, in line with what we want hemoglobin to do. When we change the pH, You'll notice if we start here around 40 again, we're going from about 78 to about 62. Again, releasing hemoglobin. Hemoglobin releasing oxygen as the pH decreases. That condition that we talked about for the developing fetus inside the mother's womb shows a clear difference in the saturation with, at any partial pressure, adult hemoglobin, which is doing the delivery, is less saturated than fetal hemoglobin. That's that alpha alpha gamma gamma hemoglobin. So what that represents is that the kinetics favors the transfer of oxygen from the adult capillaries on the uterus side to the fetal capillaries on the placenta side, meaning in the placenta, you're binding that stronger. The saturation is higher. It's carrying that oxygen out of the placenta, leaving it depleted, and favorable for the transfer from the adult capillaries on the uterus side. Now, that kind of completes our thoughts on oxygen, and I just want to say, for oxygen, oxygen is such a limiter, meaning our absolute maximum for our strength, for our endurance, depends basically on how much oxygen we can deliver, regardless of our status of conditioning. When you reach your steady state, you're running as far or as fast as you can. Maybe you can run a mile, but only a mile. But it's going to be oxygen delivered to your muscle that's going to determine those limits. Hemoglobin is a molecule that, based on all we can discover, is just about the most ideal molecule for transport of oxygen with respect to kinetics and reactivities, yields, those kinds of terms that we use to apply to chemical reactions that we use in chemistry all the time. With respect to that, hemoglobin is, carrying over 99% of the oxygen. That tells you what a strong and kind of single purpose it has. Now, in the tissues, we deliver the oxygen. Now what are we doing? We're making carbon dioxide. We need to know how that carbon dioxide gets back, because we now know respiratory. If you say it's oxygen transport, you're only telling half the story. You got to get that carbon dioxide back out, because the carbon enters the body through diet, but it leaves the body as a gas. And carbon dioxide is much more diverse in its transport mechanisms, because this hemoglobin molecule is not specialized for CO2 transport. Because it's there, we have to notice that this hemoglobin and this heme group that's in the center, one hemoglobin molecule on average will bind about one carbon dioxide molecule. You'll notice this arrow for CO2 binding is not going here to the heme groups. It's going here to kind of a corner or an edge of one of the protein chains. But the affinity is by the hemoglobin is about 23%, and CO2 is very soluble in water. So that solubility in water is about 7%. So it looks like 30% of the CO2 is going to be carried by this plasma hemoglobin system. That leaves us with 70% of the transport of CO2 to account for. When we talk about that, we want to go back to what are we really doing? CO2 plus water in the presence of carbonic anhydrase will form carbonic acid. That's missing from the middle of this. And carbonic acid breaks down to hydrogen ion and bicarbonate. So 70% is going to be involved in this bicarbonate carbonic acid system. We would think of this as dissolved CO2, and it's going to be transported from the tissues back to the lungs. Now, when we get to the lungs, we're going to reverse this, go right to left, turn it back into a gas, and breathe it out. Leaving behind water. Water has no real pH effect, or its effect is a kind of a neutralizing effect. More and more water means closer and closer to pH 7. Now we find the carbonic anhydrase in the red blood cells, so we're going to anticipate that we're going to put the CO2 into the red blood cell, bind about 23% of it to the hemoglobin, and then work on the rest to make bicarbonate. But this is that, inescapable consequence of making CO2. You're going to make hydrogen ion. So hydrogen ion's going up, and at the same time, pH is going down. So the overall idea for carbon dioxide transport, we only have to look at a couple of places. It diffuses into the bloodstream, so there's the 7% that we mentioned. We're going to transport the rest into the red blood cells themselves. Now, 93% then of the CO2 is entering the red blood cell. Here's the 23% that forms the complex with hemoglobin. And this 70% is made into carbonic acid. That's the formula right there, which dissociates into hydrogen ion and bicarbonate. Now, hydrogen ion, of course, will produce a pH effect, but it turns out that hemoglobin itself can buffer this system, removing the hydrogen ion at the tissue side. So the pH effect is moderated by the hemoglobin, and we're going to transport the bicarbonate, what's called the alkaline tide, out of the red blood cell and carry it in the plasma. Now, out in the plasma, it's going to have a buffering effect. If it runs into excess hydrogen ion, it's going to basically form carbonic acid, the reverse of this dissociation. But we have a problem when we move this bicarbonate out. We're pumping negative ions out of the red blood cell, and that quickly would produce an imbalance. So what we do is grab a hold of the chloride ion that's abundant in the material around the cells. This chloride ion is exchanged. This is called the chloride shift, keeping the charge balance in the red blood cell in balance, in equilibrium. So what we've just drawn is this right-hand picture out here in the tissues. The CO2 is produced. We transport it into the capillary. We go through the hemoglobin step, the carbonic acid step, the transport of bicarbonate, and the chloride shift to stabilize it right there in the capillaries around the tissues, and the flow of the blood pushes it on out. So we're now entering venules and veins as those vessels converge on their trip back to the lungs. When they arrive in the lungs, all we're going to do is take every one of those steps we just discussed, and we're going to reverse them. So the reason this can kind of keep working in balance is that whatever we do in the tissues, we're going to undo in the lungs. And what we're going to achieve is a net exit of CO2 and a residue that is water. So here we are, the red blood cell. The chloride shifts back out to exchange for the bicarbonate. As this bicarbonate floods into the cell, it forms carbonic acid. At the same time this is happening, hydrogen ion is being given up by the hemoglobin, and the carbonic acid generates water while it makes CO2. So huge amounts of CO2 are being generated. At the same time, hemoglobin is giving up that 23% of the CO2. So the net effect in here that we're seeing is we're making a lot of CO2. As the CO2 is transported out of the capillary and across the lining of the alveolus into this space, it's happening in such concentration that on the other side, it gasifies. It goes from dissolved in water to CO2 in air. The next time we breathe, this CO2 enrichment is significant compared to the concentration of CO2 that's actually in the air itself. So when we enrich this airspace and then exhale, it drives a lot of CO2 out of the body. Now, we've talked about the gas exchange elements, oxygen and CO2. And now we have to think about how can the body control respiration. Anytime you have a real range of metabolism, you have to think about control. I'm sitting here right now at rest, and chemoreceptors all over my body and baroreceptors are monitoring my blood, and one of those monitors is for oxygen. Oxygen and CO2 are sensed, and since oxygen supply to all my tissues is abundant right now, and CO2 is relatively low, my breathing rate is very low. I can basically breathe at rest, and I can speak easily without fatigue. However, change those conditions at all and involve a mild amount or a moderate amount of exercise all the way up to the maximum aerobic exertion. And we're going to make wholesale changes around our body to make maximum use of this respiratory system. We're going to find local control within the lungs for the gas flow itself. Are we going to direct inspiration air to a particular alveolus or not? We're going to control that through sphincter muscles, smooth muscles, that are around the arterioles leading to the alveoli in the lungs. We're going to monitor the overall effect of that gas flow and oxygen uptake and CO2 release by monitoring the blood around the body. And a great place to monitor the blood is at the base of the brain. That's going to put an overlayer of central nervous system control that can affect everything from heart action to lung and breathing rate. We talk about ventilation, providing the flow of air in and out of the lungs, with perfusion, the exchange of gases in capillaries, and the different conditions for oxygen and CO2 that occur in the lungs and in the tissues. We're going to adjust ventilation at the alveoli, where the exchange of gases is occurring, the perfusion of the alveolar capillaries. This is accomplished by local control. Now, that means the central nervous system is really not controlling that. Basically, the conditions inside the lungs are controlling them. So if we reduce ventilation, so we have excessive perfusion, these arterioles will constrict and reduce the flow to this alveolar sac. We talked about the alveolus looking like a bunch of grapes. Well, this is an individual exchange sac. If we enhance ventilation and we don't have adequate perfusion, we're going to basically relax those sphincter muscles and allow those arterioles to dilate and basically direct blood into that because there's more capacity for perfusion. So the conclusion to remember from this kind of simple comparison is that blood is redirected to the alveoli with more oxygen. So more oxygen, that's where we're going to send the blood in the capillaries and the walls of the alveolar sacs. Air is going to be directed to the alveoli with more CO2. So we're going to reduce ventilation, and if perfusion is excessive, then we're going to increase airflow to those that have more CO2. So blood where there's more oxygen, and air where there's more CO2. Central control is accomplished by those respiratory centers we named last semester when we were talking about the central nervous system. Here's that brain stem, and it's the brain stem all the way down to the foramen magnum. But here we have a couple of centers we call the rhythmicity center and the medulla, where pH sensors are located. We have centers for these central nervous system controls in the pons and in the medulla oblongata, which set our basic rhythm, our basic rate to control our breathing. Now, these pH sensors are going to be monitoring the cerebrospinal fluid, and isn't that interesting? So we're working to oxygenate the blood, but our life is really our central nervous system, especially our brain. Under no conditions does blood flow to the brain diminish. For the size of the organ, it's relatively high, and it stays fairly constant no matter how active we are. But this is really monitoring where the rubber meets the road, how much oxygen you're getting across the blood-brain barrier and into that cerebrospinal fluid in order to sustain the operation of nervous function in the brain. So from higher centers up in the cerebral cortex and the so-called apneistic and pneumotaxic centers in the brain stem, we're going to control exhalation and inhalation, breathing rate, and the respiratory rhythmicity through these brain stem respiratory centers. The peripheral nervous system, however, is not left out of this. There are sensors around the body that basically are there to detect inadequate levels of oxygen or abundance of CO2. The carotid body has sensors in the carotid artery. Now, this is the main artery departing from the aorta and from the brachiocephalic trunk to supply the main blood flow to the brain. So in the carotid body, if we decrease pH or oxygen, or we increase carbon dioxide, those are detected in the carotid body. They are wired to the glossopharyngeal nerve, one of the cranial nerves, and they transport that information to the brainstem. The aorta is a great place to monitor blood. When we work to oxygenate the blood, the richest blood in the body with respect to oxygen should be in the aorta itself. I mean, we noticed that the systemic part of the systemic circuit that we call the cardiac circulation takes its blood right out of here at the base of the aorta. Normally, we only CO2 sensors are active in the aorta, and the signals can go directly to these intercostal muscles from those sensors. Another example of local control. And also to the diaphragm. So the major breathing muscles for moving those lungs can signal directly from those sensors, basically affecting the depth and the rate of respiration and ventilation. So here we have the cycle that we all go through every day from at rest. This exercise, where we're beginning here, produces higher CO2 and lower pH. The brain senses that lower pH and it accelerates our breathing. We breathe faster and deeper during our exercise. We are going to purge that CO2 as we exercise. When we stop, of course, we breathe hard for a while. And the main issue is, when are we going to restore our CO2 balance to normal? And then we can slow down the CO2 loss and oxygen increase to match our at rest level of exertion. rate of respiration is controlled by CO2 in the blood more profoundly than oxygen. It is possible to hyperventilate if you breathe deeply and rapidly for a long time. You do get a little dizzy. Hypoventilation can produce a kind of a sense of lightheadedness or even fainting from underventilation. And these controls that we just discussed will maintain that balance. Myself, I can remember a time when smoking there was a positive PR message for smoking. Absolutely positive message. In the 1950s, Mickey Mantle, a very popular baseball player for the New York Yankees, sort of the darling of the time, would advertise cigarettes in all the major publications. I can remember when the Surgeon General's report for smoking came out, the first massive collection of data on the effects of smoking. Now, this is something you've lived with your entire life. Smoking for the period of your life is a negative. No amount of smoke is good for you. However, we have seen a rather interesting effect of this data on the public's behavior. And it's another example, like the COVID-19 epidemic, of science versus social. This one is very clear that no amount of smoke is good for you, even secondhand smoke, so that if someone who chooses to smoke in a restaurant will be asked to leave or should be asked to leave, to keep secondhand smoke away from other diners. The data is reinforced by pictures like you see here, a smoker's lung and a non-smoker's lung, comparing blackened, tarry, contaminated tissue with clean. That pink color, that white color tells you it's more the material that makes up the lungs that is showing. The statistics and data, and by the way, these are now old. I made this slide so long ago that these are certainly the numbers. They may be larger. We estimate that 50 Americans die every hour, 440,000 every year from smoking. The critical thing about smoking, though, is that it stresses organs other than the lungs. And so every mortality factor that we know of, whether it affects the heart or the kidneys or other systems, of the cancers that can occur at any location in the body, they are all increase those mortality rates by smoking. So this is actually clearer than COVID. When you look at this number, 443,000 every year is what smoking is causing. Now, today I think I saw a number for the United States, 500,000 cases of COVID-19 have been registered. We are over 500,000 cases and 125,000 deaths. So we aren't nearly at a year yet, but you can kind of compare those numbers. This is a number that, based on our current behavior, is going to increase and repeat every year. The effects on the body begin with the effects on the cells. We've talked about a respiratory system that's a cul-de-sac down and back, and we have to have the ability to keep that clean. And it is smoking, it is the contamination from smoke that causes the problem. There are combustion products, gases where the harmful molecule is free as a gas. there are residue-leaving molecules, those things that are called tars and nicotines that basically can coat the internal part of the lungs and have bad effects on the cells. But there's also particulate matter in smoke. When you blow smoke out and you get that big white plume, you're actually putting out a very fine ash. And it is the ash that's accumulating there. So what happens throughout the respiratory system? What we see with smoking is the cilia, such as those on your mucus escalator, slow down, then they stop working, then they're killed. So the lining of your respiratory tract, especially your trachea and bronchi, are basically now lined with dead cilia. Fewer cilia means your mucus escalator basically stops. You know what happens at the mall when the escalator stops, you have to walk up the stairs. Well, there's no such possibility. You only have coughing. So the smoker's cough increases, and the frequency and the severity of smoking cough does increase. That produces a stress on the respiratory system. Coughing is a explosive effect, and so whereas it's a normal activity, it only retains its normalcy under a normal amount of coughing. Repeated coughing causes those muscles and the connective tissues around them to reinforce themselves, to get stronger and more forceful, putting more air pressure on the whole respiratory tract. That means they become stiff and less rigid. And so what happens as a normal cough in a non-smoker becomes dangerous repeated activity in the smoker. As this happens, the goblet cells pour out more mucus because there's a contamination. Mucus is actually a nutrient. If you don't move it, things grow in it, and it causes more infections in the lower respiratory tract. That coughing can irritate the bronchi all the way from the bronchioles, the secondary bronchioles, all the way up through the throat and mouth. The walls of the bronchi thicken due to less elastic tissue and more stiffening. And that repeated pressure expands the alveoli and causes them to stiffen. The secondary effects of coating them with smoke, it's sort of like painting them with varnish. It stiffens the walls. So these stiffened alveoli begin to rupture. And what you want to compare here, this goes back to our definition of compliance. You see in the healthy lung on the left, the literally countless numbers of very, very tiny air sacs out here in the lungs. Whereas what are we developing here? Larger and larger holes. As the walls of those alveoli break down, you lose surface area. It's easier to fill that big cavity than it is to draw air into the small alveoli. So compliance goes up, but exhaling that air, pushing it out, is much less efficient. So this is the condition we call emphysema, almost always linked with smoking. And prior to death, in its advanced stages, requiring oxygen enrichment because the lungs no longer can draw enough oxygen out of 20% air. You need to enrich it with pure oxygen in order to keep the oxygen levels at a safe level. This is strictly a physical effect. You can look at this. This is basically a problem in solid geometry. Here's a cube with 30 micrometers on a side. The surface area is 5,400 square micrometers. And since surface area relates to transport of molecules, that's the limit for transport in this larger cube. Take this and divide each dimension into 10 micrometer cubes. So here's the bunches of grapes. You have 27 small cubes, and you have cross wall cubes, these vertical walls and these horizontal walls that run front to back in this drawing. That increases the surface area to 16,200 micrometers, about 3 times the total surface area. Now, you have to mount your capillaries in the surface area. So 3 times the area over here if you retain the original alveolar structure, a surface area effect. COPD, or chronic obstructive pulmonary disorder, can be caused by a number of factors. Chronic bronchitis, coughing, if there's a bronchial irritation. Emphysema can be accompanied by pulmonary hypertension. We're talking there about basically blood pressure to the lungs being abnormally high. And the reason is your oxygen sensors are saying, whoa, not enough oxygen. And they're doing what you do when you start running. They're basically speeding up your breathing and speeding up your heart rate. Cor pulmonale is an enlargement and a strain on the right side of the heart, the side that pumps to the lungs. Also, this will cause a erythropoietin release and an increase in the hematocrit as the blood tries to adjust. Decreased oxygen tension, just like moving to high altitude, but in this case due to a smoking-related reduction in surface area in the lungs. Tar is a deadly substance that comes from the combustion products. 70% of what is inhaled is deposited. Now your alveoli macrophages are going crazy trying to scavenge that dirt out. Now, non-smokers have to deal with dust and pollen and other things. Smoke, smoke that they walk through as they're walking down the sidewalk. But what a smoker is doing is putting 20 to 40 to 60 cigarettes worth of that smoke into the lungs every day. So these alveolar macrophages become overwhelmed, they start dying. You're more subject to lung infections. Carcinogens are mutagens that can cause cells to mutate. Basically, carcinogens are those mutagens that are associated with the risk of cancer. Every carcinogen is a mutagen, and as this left bronchus, as you see here, this undifferentiated tissue, this lump, has grown to a size where it's essentially cutting off or restricting airflow to the left bronchus. So what we have here is you may in fact be able to work hard and inflate this left lung, but inflating the left lung takes a lot more effort and a lot more time. The presence of this tumor is blocking the airflow. Good place for a forced minute value. Lung cancer is 85% more prevalent in smokers than in non-smokers. We keep showing you the pictures, red and healthy and black and more or less shrunken. Showing you the real pictures kind of shows you the amount of deposition of tar that you had inside. Exchange areas are related to these alveolar sacs. I've seen various estimates. The one that impressed me the most was from a general biology text where they said if you could actually take each of these as a sphere and kind of take the holes out but calculate the surface area in all the alveoli in a normal person's lungs, in other words, the area were laid out flat, the surface area in a normal person's lung would just about cover a tennis court. That's how much surface area you have. It kind of shows the selection's been very strong. Why do we have that so much? Because oxygen supply is so vital to our performance. We have nicotine, a deadly poison. It causes constriction of blood vessels. It raises blood pressure by itself. A combustion product of carbon monoxide. Carbon monoxide binds hemoglobin tighter than oxygen, and so therefore affects the amount of oxygen delivery. The highest single cause of deaths related to smoking, it's no surprise. We have so many diseases, not just smoking. that put a strain on the heart, and it is the heart that fails. And true of smoking as well. Most of the deaths are related to cardiovascular failure. So when you look at cancer statistics, you look at observed deaths and expected deaths and what's called excess deaths. So when you expect this number but you observe this number is extra, over and above the top. So the percentage of the excess, the relative death rate is 1.5 over a non-smoker when you're a smoker. When you break it down, you'll notice some numbers that are dramatic. Coronary artery disease, 1.7 times the probability or the possibility of that disease in a non-smoker. You'll see down here, cancer of the bladder, twice. Cancer of the mouth, larynx, or esophagus, the respiratory tract, 5 times. Gastric and duodenal ulcer, 4 times. Lung cancer, 10 times the probability of death, 10 times excess deaths as a result of smoking. Now, this is now such a well-known statistic. It keeps being tested. You can bet that all the money the tobacco companies had, that they put out all of their moderating statements that they could, but the government and the medical community has come out and said no amount of it is good for you. We do have an observation on this blue line. Notice, starting at age 25, there's a decline in lung function. That's normal. So at the age of 75 and maybe 80, up here, what we're observing is a decline to maybe 70% lung capacity. But that's still a very usable amount of lung capacity. And look at the gap between the line that represents disability. Now, if you look at the slope of that never smoked line, and you look at the smoke of the line of the regular smoker, see how much more quickly it dips. If you stop smoking, you stop declining on the solid line slope, and you basically decline on a slower slope. So this dotted line at the top stopped at age 45 and basically didn't reach disability until maybe, what would that be, 90? So stopping smoking is a good thing, but notice it's still declining. It's declining at the normal rate of decline. If you stop at age 65 or stop at the level of disability, you'll notice one thing. These curves never go back up. The lungs that you have at 25 are the best you're going to have, and they're going to decline slowly. By the time you're 50, your lungs are still working at very, very high level if you don't smoke. Life expectancy, this is one of those medical statistics that come from big data. And there's no doubt that it reduces your life expectancy. The average at the time I did this slide was 12 years, or a reduction in your life expectancy based on smoking. So, oops, sorry. That's the end of the respiratory chapter.